All right, welcome back. Today we're going to look at the last exploration from Unit 6, Lesson 2. This is Exploration 3, and we are going to look at the second part of our protein synthesis process. In Lesson 2, we've been talking about this idea of protein synthesis where the information found in DNA is copied and converted into a physical structure called a protein. We talked in Exploration 2 about how the first stage of this process is called transcription because we are transcribing a copy of the DNA into a temporary RNA molecule called messenger RNA. In this exploration, we're going to look at translation, which is what happens when your cells take that messenger RNA, that copy, and they use it to build the protein that was needed in the first place. All right, so let's take a look at this. First of all, it's important to know that in the world of building proteins, there are only 20 ingredients. We call those ingredients amino acids. Those 20 amino acids, some of them are found in our bodies naturally. Others of them we have to get from the food that we eat. But all of the proteins that your body needs can be made from combinations of these 20 amino acids. Now, that might not sound very much, but one way that I think about it is like a chef. If you were to give a chef 20 basic ingredients, 20 kind of stock ingredients, how many different types of food could they create? I mean, the possibilities would almost be endless. And it's kind of the same way with proteins. From these 20 amino acids, we combine them in different quantities and in different orders to create those physical structures that our body needs. All right. Now again, in this case, we are looking at translation. Translation is where we are going to translate literally between the language of nucleic acids to the language of proteins. And we do this through a series of a couple steps, all involving different types of RNA. All right, so we looked at how the messenger RNA, or the mRNA, is responsible for copying the DNA and bringing it into the cytoplasm. In the cytoplasm, we have structures called ribosomes. Now, we first looked at ribosomes last semester. We said that ribosomes are responsible for building proteins, and we left it at that. In this exploration, we're going to look at how those ribosomes actually do that. Now, ribosomes themselves are another type of RNA. We call it ribosomal RNA. And so when the messenger RNA leaves the nucleus, the ribosomes are able to see it. They recognize that something needs to be built, and they attach themselves to that messenger RNA to begin this translation process. Now, we also talked last semester about how that protein is built, and then after it's built, it has to be packaged up. We're not going to talk a lot about that right now, but here's a section to describe kind of the review of the idea that after proteins are made, they are wrapped in a vesicle by the Golgi apparatus so that those proteins can be transported wherever they need to go. All right, again, don't skip over these graphic boxes. They're really good. If you need to pause the video to make sure that you get this correct, I would do that. But make sure that you go through these. There's a couple of these in this exploration. These provide a good summary of the key events that are taking place in this protein synthesis process. All right. Let's talk about amino acids. Now, we said that there are 20 different amino acids and that combinations of those amino acids are what produce our proteins. But those amino acids all have a specific code. Now, the code for an amino acid is actually called a codon. Works out pretty well. And a codon is determined by a 3-letter combination. When we look at our messenger RNA molecule, every 3 nitrogen bases, every 3 nucleotides, gives a code for one type of amino acid. In this case, CGA is the code for the amino acid arginine. UAC is the code for the amino acid tyrosine. And remember, there's 20 different amino acids. Now, the way that it works out, and I'll show you this in a second, there's actually a few ways to code for the same amino acid. In other words, of the 20 amino acids, a couple of them can get made by a couple of different codes. And this is going to be important later when we talk about mutations. But for the most part, each amino acid has its own specific combination of 3 letters that we call a codon, and that codon is what determines what corresponding amino acid goes with it. Now let's look at how this gets built. Real quick though, before we do that, this is a codon chart. Now there's a couple of different types of charts. This is the circular version of the chart. And in order to figure out which amino acid goes with each codon, the scientists have created a chart like this to help with that process. So in other words, here's how you use it. The first letter of the codon is represented by this large inner circle. The next letter of the codon would be this next quadrant. And then within that, any of these remaining letters would be the final and third piece of that codon. So let's just say you had the codon GGG. If your codon was GGG, you'd start here, you'd limit yourself to this quadrant, to this section of the quadrant, and then to that letter. Now in this case, this is what I was talking about before, where there's actually 4 different ways to get to glycine, which is this code right there, glycine. If you had GGG, you would get to glycine. But actually, any codon that starts with 2 G's is going to give you the same result. And again, this will be important when we talk about mutations, but here's how you read that chart. First letter, second letter, and then third letter tells you where you need to go. All right. All right, let's look at how this gets built. Here are the steps of translation. Now, as we said before, once the messenger RNA leaves the nucleus, it is attached to by a ribosome. Now, ribosomes actually have what they call a large and a small subunit. In other words, kind of like two parts of a clamp. The large part of the clamp is going to hold on to the protein as it gets built. The smaller part of the clamp is going to hold on to the messenger RNA as it gets read. All right. Now, we've talked about messenger RNA and we've talked about ribosomal RNA. There's a third type of RNA that we introduced to you earlier called transport RNA. And the transport RNA has a very important job that is just what its name would imply. It brings the amino acids to the ribosomes to be put together. Because remember, on the ribosomes, on the messenger RNA, are those 3-letter codons. Each codon goes with a specific amino acid, and each specific amino acid has a specific tRNA that brings it in order to be built. All right, now on that tRNA, the corresponding code that goes with the codon, the 3-letter combination on the tRNA is actually called an anticodon. The anticodon is what matches up with the codon to be able to get the proper RNA, or the proper amino acid in place. All right, so let's look at what that looks. Let's take a look at what that looks like. This animation is pretty good as far as describing this process. So here again, here are 3 parts. Here are 3 types of RNA. We have our messenger RNA, which is right here. We have our ribosomal RNA, which is this green structure, and again, it has a large piece and a small piece, kind of that, like we said, a clamp, large clamp and a small clamp. And when that large clamp and small clamp attach themselves to this messenger RNA, they begin to read the message 3 letters at a time. Okay? And again, every 3 letters is what we call a codon. The codon matches up with a tRNA, which is this structure right here. It's called a tRNA for a lot of reasons. Number 1, it transports amino acids. But number 2, I think people think it looks like a T, kind of. I don't know. It looks more like a backward, almost like a 7 to me. But the tRNA, as you can see, has complementary anticodons attached to it. And when those complementary anticodons match up, they bring with it the amino acid. So let's back up a little bit. Right here. Our first tRNA is going to come in, it's going to click in place, and once it clicks in place, the amino acid that came with it is going to pop off. Here you can see the second transfer RNA kind of bringing in. As we shift down 3 more letters, the next tRNA clicks into place. And when it does that, once again, it releases the amino acid that it brought, and those 2 amino acids then connect together. This forms what we call a peptide bond. Now, as we keep on moving down the messenger RNA, the old tRNAs leave, and they leave to go pick up new amino acids. The new tRNAs continue to come in, they click into place, and then they release their amino acid that came with them. Until finally, what we have is we get to our last codon. UAG is what we call a stop codon. Once we get to that stop codon, no new tRNAs are coming in. Our protein chain that we've now built gets released because it's finished, and this ribosomal structure can go find a new messenger RNA to begin building a new protein. All right, so once again, here's a diagram. Highly recommend you go through, click the right answers on it. That way you can go back and review. But again, this process, something you might want to go through a couple times. Again, it's very narrative driven though. So if you can click through these pictures and you can talk about what's going on, then that's a pretty good indication that you understand how protein synthesis works, which is obviously the goal. All right. Last thing we're going to talk about is mutations. Now, real quick, before we get to mutations, I'm not going to show you this video because you can go back and watch it on your own, but I would highly recommend that you go back and watch this simulation because it's really, really good at demonstrating how this process works from start to finish. Again, if you're having trouble understanding, go back through, watch this video a few times and try to pick out those key parts that are accomplishing the different functions of this process of protein synthesis. All right, let's look at the last thing. We mentioned earlier that mutations are something that is going to happen in this process. We mentioned the idea of mutations earlier when it comes to replication, DNA replication. But a mutation is any time a sequence of DNA is changed. Anytime there is a mistake, anytime something is different than what it's supposed to be. And it can happen during replication or it can happen during protein synthesis. A mutation during protein synthesis usually means that when the messenger RNA was created, it didn't quite copy down the DNA perfectly. Now, here's the thing. It might not matter because when the ribosome goes to read that message, as we demonstrated earlier, if the 3rd letter of that codon is changed, might not make a difference. In some cases, it certainly will, but it's possible that a mutation in the 3rd part of the codon might not change the amino acid at all. However, if you were to make a mistake on the 1st or 2nd letter of a codon, then there's almost a certainty that the wrong amino acid would be added. Now you might wonder, how big of a deal is it for the wrong amino acid to be added? And it's kind of like, once again, going back to cooking. If you have a recipe that calls for sugar, and instead of adding sugar, you add baking powder. Well, your cookies or whatever you're baking are going to taste dramatically different if you add in, a cup of baking powder as opposed to a cup of sugar. And the same is true with proteins. Even one wrong amino acid could totally throw off the structure and function of that protein. And so it's very important that this process is carefully regulated so that there are as few mistakes as possible. All right. Now that takes us to the end of this process. Again, highly recommend that you go back, you read through these sections, you make sure your notes match up with my videos. And again, this animation right here is fantastic. So if you didn't pause the video, maybe go back in your online textbook, watch this video, watch this animation, and talk to yourself about what's happening at each of the stages in this process.